as a leader, uh, president of the society, very active, very uh, immersive kind of president. Sometimes president is decorative positions in societies or associations, but he is always embedded professor and president of the society. And you all know he has been a very long career as a professor and head of the department till he retired. Department of Pharmacology and another premier institution, national institution, that is All India Institute of Medical Science in New Delhi. And for 2 years, as I mentioned, he was also director selected by Government of India to lead the institutions that Indian Institute of Toxicology Research at uh, Lucknow. He was also president of Society of Toxicology. I was president there for 2 terms, and so that way Gupta is well known, and for some time he was also officiating director of AIMS and also a spokesperson during some turbulent and controversial time. He was all the time coming on television, and Dr. Gupta is also members or chairman of many societies or government committees which deal with the GLP, good laboratory practices, and also he is also member of many DST, Indian Council of Medical Research, and DVT, whatnot, and president of governing councils of Indian All India Institute of Medical Sciences, many one. And currently he's chairman of the review committee on genetic manipulation, and has guided many research scholars for their PhD or master's degree, MD or MSc. Gufran also worked under him when he was a student of Jamia Hamdard, MSc Clinical Research. He completed internship under him. So, and currently also he's earlier for after his superannuation, he was associated with the Translational Health Sciences Research Institute of DBT in Faridabad, and currently he's the senior advisor of a WHO committee. And we are very happy to have you here this morning, sir, and we are privileged to that you kindly agreed to deliver and enlighten us with your talk, which have been always provocative, and also milestones for us in many senses. So thank you very much, sir. So I invite Professor Y.K. Gupta to have the first talk of the day. Professor Y.K. Gupta, sir, please.
Good morning to all of you and namaskar. My deep regards to Professor Akbar Shah, who is not only the president of this society, but this is his brainchild, I would say. His brainchild and then child, which he has nurtured and which has now become almost, maybe after a few years, it will be decade old. And my regards to Dr. Raisuddin, who was also one of the, I would say, the pillar of this society when it was framed, when it was made. And I can see Ankita also, and she is another pillar, and of course, Dr. Ziaur Rahman. It's a matter of always pride and privilege to come and speak in this university, which is known for its academic excellence, I think, ever since this university came into being. And it is, I am fortunate that I have been here once upon a time before also in this very auditorium. And what a nostalgic feeling, Dr. Ziaur, which you provided to all of us, in addition to hosting this important meeting and conference. So my salute to you and to your entire team, and to the support which this university gave to this meeting and this conference. My co-chair, Dr. Kurian, I am told that I never knew him before, but I am told that you are one of the top ranking regulator of the country, and that's what something which is very important, because whenever we talk of anything which is alternate to animal experimentation, we always look for what is the gap between what we do in the lab and what is the gap when we want to introduce into the regulation. So I think that's this strategically this co-chair and chair, and both the chairs are very important for me strategically, I would say, on academically as well as strategically. And I also apologize on behalf of myself and Dr. Raisuddin, because we tried to start from Delhi at 4 o'clock, but it got delayed. And then there was some issue in the between, and then you had to wait for some time. Having said this we may like to understand. Maybe Dr. Raisuddin and Dr. Akbar Shah will give you, and Dr. Zaur will talk to you later why and this conference, what was the need of this society. The entire word in the biomedical sciences, and of course I can say Adib, I will not, I will feel guilty if I don't mention Adib's name. Because these are the founders. They have been looking, the entire world has been looking for the rationality of the use of animals in the generation of evidence for safety and toxicity. And when we say so, when I was doing my MD in 1970s, I remember then I used to catch a dog, and catching a dog for just doing a vomiting experiment. And if I remember that, had this dog been my own pet, I would have wept 10 times before I do that experiment. That was, but then we enjoyed at that time, because that was an excitement. That was a requirement at that time. That was a regulatory requirement. That was a requirement of this is. And I also remember then when we used to do the experiments on the frog, catching the frog was an enjoyment, because it used to help our girlfriend, because they never used to catch the frog because it used to slip. And this was the prerogative of boys to catch the frog and give it to them and then expect thanks from that. Thanks, getting thanks from a girl at that time used to be a big thing for us. But when this frog was pithed, for which we also helped her, if you remember those boys, I don't remember that you have ever pithed a dog. And when this frog used to cry, and then you just used to unnecessarily sometimes, but maybe it was useful. I used to argue for also, and then you throw it. And when you used to throw it, then even the half conscious frog, its open abdomen, I think if you just remember that scene, it was a pathetic scene. And you just remember, had it been your own body, how you would feel, because they cannot say, they cannot feel, they feel, but they cannot say, they cannot express that anguish. We used to do whatever it is. And even today, when we do clinical experiments, clinical research, the common proverb was, in India, the human are being used as a guinea pig. Why is guinea pig word is being used? Why not just say that elephant or human are used as a tigers? Because research is done not only in guinea pig, research is done in from dog, monkeys, anything. But human are being used as in India for clinical trial. The Western world is using human as a guinea pig. The reason is only, how many of you have seen guinea pigs? I think there is nobody who has not seen it. The simplest, most docile animal who has complete trust on you, and he will always smile even if you kill him. And that's why he becomes the sufferer. That's why people say guinea pig syndrome means you use anybody who is most docile and a most cruel experimentation of cruel behavior. That's not acceptable now. And then we look into that. Yes, fine. Science is one. Generation of evidence is important for science. Generation is important, particularly for safety and efficacy, because ultimately this will save the lives of millions of people if it comes into the practice, whether it is a drug, whether it is a chemical, whether it's a device. But the important thing is, for generation of that evidence, is this the only method, or there can be any other method without sacrificing these innocent animals? Can that method be validated and replace this? Does not mean that we are completely replacing it, but we are saying as and where possible. That's why the philosophy of 3R came. 3R is not a big deal, but 3R, reduce, refine, and repurpose, or whatever R's are there now. We have made several R's for that purpose. It's just, you are not saying that you finish it, but you reduce it as and where possible. Refine it. When you say refine it, that you are doing a blood pressure experiment, only blood pressure experiment, and then you throw it the animal. No, it's not the way which you used to do in 1970s for our thesis. But if you are putting an experiment, can you use all sort of instrumentation with this? See what is the blood pressure, what is the heart rate, see what the biochemical changes, see what is the EEG changes, and see what are the connectivity with the different organs, and you generate as much as information which otherwise would have required more than 200 times. You generate from one. And just doing that, then you develop algorithm, and then you postulate, and then you predict that yes, I do not require 8 animals or 10 animals. I can do by algorithm, and then I can produce this results. This is the algorithm which is artificial intelligence based, and therefore minimum number of experiments are required now. And that's why what is called as a refinement of the techniques. Refinement of the techniques can also be in anesthesia, where you used to put the pithing the needle most cruel way, can do much more simpler refinement with the instruments, with the gadgets, with the calculated anesthesia, which will be happy to the animal will not be feeling bad. At least it will be control anesthesia, things like that. The surgical process has also become much. So you refine the technique, you refine the generation of the data, and thereby reduce the number of the animals who are important and where it is must to be used. The other thing what we say is the reduce the number, because if I ask many of those who have passed MD or MSc or even the PhD, what is the minimum number of experiments you require? What is the N for a group? Everybody will say 6 to 8 animals. I ask 6 to 8 animals, why? From where the 6 to 8 animal has come? Hardly anybody knows the background of this. अरे भाई 6 animal क्यों चाहिए? Then if you just know that these 6 animals are required because the genetic variation between these 6 animals, which are inbred animals, we are supposed to have these inbred animals, not what you have in the even in the from the lab, which are not inbred. And means 7 generations of brothers and sisters are mated together in rats or mice. That is called the inbred. Hardly anywhere they use inbred animals in practice, but they write everywhere. And then the genetic variation is minimal, and therefore 6 animals are enough to represent the variation. Now, if the 6 animals are not required because now you have a genetic genomic variation minimized, then maybe 4 animals may be required. Maybe if you have generated that evidence in vitro or in cells or maybe in experimental, like in chemicals, therefore you have to just validate some purpose you require it. So you reduce the number of animals, you reduce the number of groups, and that requires a careful consideration. And that is, but then dichotomy comes where the ethics committee, when the animal ethics committee members evaluate a project, then they think that their role is only to reduce the number. I think that is also unethical, because if the required information is if the required information is to be taken from the 6 animals or 4 animals, Giving 3 animals is a criminal, because 3 animals will also go waste. And giving 12 animals is also criminal, because that will also be grossly wasted. So that is the optimum use of animals is important. I remember the rehabilitation is so important. I will just give you an example when in fact this was happened with me. I used to put intracerebroventricular chronic ICV cannula in the dog, and after the dog is experiment is over, take out the cannula and then put the stitch back and they release this dog. He never realized that this healing has not taken place and this dog is crying in pain and hitting his head to the wall. Then once upon a time, and once at a time, my teacher, Professor K.P. Bhargava, he called me, "Is this your dog?" I said, "Yes." He said, come to me with this dog to my room." I did not understand what is the reason why he called me in his office, which is one of the five-star hotel at that time, with dog. And I could catch the dog. And then he did not say anything. He just took a gauze piece, a saline, and a antiseptic and cream, and he just, I got the message. And that was the message without conveying any, he did not suspend me. He did not say anything, just he did that. And that is where I think the sense of gratitude to the animals will develop, should come. Lastly, when I saw I lost my dog, one of my pet dog, and I saw after 3 days that dog was on the same table when I used to do that experiment. Because this dog was going around and somebody caught this dog and sold it for 16 rupees to one of my colleagues, and this dog was on the table. I rescued it. So that is what I say, why we look for, can there be alternative system for animals for biomedical research, for which can provide a legitimate evidence of safety and efficacy without compromising the science. I always believe that we are not saying that you stop it completely. No, that's not the way. Because where it is regulatory point of view is required, you have to do it. But then the ultimate objective is we develop a model, we validate that, go to the regulator and say that, yeah, there is a model which is equally effective, equally sensitive, and equally validated, and therefore this model should be replaced. That is our objective of this society. And I salute the people who thought about it. And this is the first of its kind society, which very difficult to survive such societies. Why? This this creates ripples in the society. This cannot be funded by industry. Hardly any industry will fund it except Adib, people like that who are really involved. It is, and therefore, and it's also not so attractive because you can see the attendance also with as compared to any clinical society, things like that. So I must say my greetings, and in last 5-6 years we have been able to do something tangible also, influencing government. Wherever you talk, you can talk about this, and then partly the system will work. It's very difficult to move a regulator, very difficult to move, because they're not supposed to move. They are supposed to toe the lines which are written. That's the Bible. But then to tell them that this is a regulatory science and therefore this needs to be altered is a responsibility of society like this and scientists like this. So that is what is important for this. And that's why I emotionally touched with this society and contribution to all. So I would just put a few slides. My talk is basically gist is over, but I will just talk about what are the alternate need, challenges, and strategy. And in this case, why I say strategy is important, because these type of changes cannot be done by just raising a flag and taking a flag march. No, it has to be strategically, because you have to involve the regulator, you have to involve the policymaker, you have to involve the fellow scientists, you have to involve the industry, because you have to involve the funding agencies, and you have to connect with the international bodies, because what in USA annual model, alternate model is developed, if you develop, validate it, and you have to last the most important, which is least happening in India, inter-laboratory collaboration. animal models. I was doing an exercise. All universities are working on alternate. Several IITR, JRF Research Foundation, and IITs, several medical institutions, several pharmacy institutions, but there is hardly any network. And of course, the institution which Dr. Akbar Shah is heading for many years, but there is not much connectivity between us. And there is, but through this society, we think that this connectivity can, through this conference, the connectivity can increase, because you require a validation which I have done in my lab. It's useless if I say that I have developed a model of this plant, and I can see that this plant is a photosynthesis alteration. I can see that what is the skin sensitization check, whether it is done or not. ITRC has developed a model, but has it been validated by other models? Has it been validated by CRO like JRF? And then if it is validated, then has it somebody approached to the regulator, the government, to include this? That is why the collaboration or interneting is important. So this is I would say, the core dimension when it says science, sentiments, ethics, and alternatives. Science, sentiments, and ethics. This is what we have talked about. So sentiments, and the answer is, for animal experimentation, the fourth dimension is alternatives. That's what. And I again say that we have to be very careful when we cannot be activists. I used to say, the earlier representative of PETA and also Ankita, that we have to be a bit balanced. At least we have to appear balanced. We cannot say, "Band karo animal, yaar, nahi to jhanda leke." Jise just to quote, in UK, my guide Feldberg and Borison was just close to Nobel. उनकी पूरी lab जला दी वहाँ के activists ने, because they used to work with dogs. That's not the way. Did it stop? No. Did it stop animal? But it created ripple. So you say that yes, we are promoting the alternate to animals wherever possible. That's what it is. That's what important thing. So now it's still important. See, why experiment? The biological similarity, short half and controlled atmosphere, susceptible to human disease, and people argue. In fact, I also argued on that the vaccines could not be developed, transplantation could not be developed unless there has been animal experimentation. But the other is, why these experiments not? Are they futile? Wherever they are futile, must question. Wherever they are painful, must reduce it. And where they were done with consenting animals, because you don't have a consenting animal. So that is what is important. Now, where the animal testing is used today, basic and translational research, even basic research, but now the basic research much is being reduced because of the even organ on chips, organoids, and artificial intelligence, the algorithm development. So you have to just think about it. Pharmaceutical research and development, that is one area where we need to convince the regulator that these are the 20 areas: acute, subacute, chronic, carcinogenicity, mutagenicity. If you just see OECD guidelines, how many animal experimentation are to be done? Can we reduce them? Yesterday, we had 2 days back, we had a meeting and chairing the GLP next technical committee, and as we said, I will forward that list once it is finalized, that these are the scopes, these are the test items where the alternate animals can be considered if they are brought to the notice of the GLP technical committee. You develop those models. So, toxicity testing, animals and chemicals, and these are the mandated. Now, what industry will do? They cannot switch over to animal alternate, because this is regulatory wise, it is required. So this change is required, and this can be done gradually. The issues with the testing animals. Now you see the validity of animals and ethical issues. Now you see that these are the common things: replacement, I have talked about, reduction, refinement, and another thing which CPCSE or CCSEA has agreed is rehabilitation. Rehabilitation of whether monkey, mouse, rat, once they use it. And then the another R which we have introduced, we conventionally call it, is reduced cost. That is also important. If you use the larger animals as versus compared to zebrafish, you can generate the reduction in the cost. Reproducibility, because the variability in the species or the interspecies animals is much less. And rapidity. If I do a zebra test, neurotoxicity can be done in just a 1 month time, where if I do in monkeys, it will take 6 months, which is better because it has to be validated. That's important. And the outcome and new alternative methods, but not expensive of, but not at the expense of the human. Why I'm saying this last sentence, not at the expense of the human safety, because you have to ensure that the result which you are getting because of avoiding the larger animals by using this non in vitro, you are not compromising the safety. That means you have to validate this is as good as the other one. That otherwise it will not be. This is something would you rather see live? Now, this is something which you have a wonderful here. Your pet animal, maybe pet, maybe here you can see rat, or this is the slide which my one of my colleague put it. यार आप तो बात तो बहुत करते हो. एक आपके पास ये कुत्ता है, यहाँ पे हमने चूहा बना दिया. ये आपकी डॉटर, ग्रैंड डॉटर. Which you will like to say? And how many of you will say? Because the drug which is done for safety analysis on this, if it is not valid, then there is a risk for the life, because this drug will be used for the child. So which you will prefer, whether this or this? Raise your hand. Naturally, nobody will say, I, I will like to. Can, then the next question, can both thrive? Can both? And the answer is, are there any alternative? And that's why the answer is yes. Big yes. Both can live together. And that's why the alternate. And that is the basic philosophy of that. The only one answer. I don't have two answers for that. No, this you can do, but this, but this, but this. But only thing is that if you have an alternate, and that's what purpose of this thing. And what are alternatives? I will not go into this detail. The strategy is that when these alternatives are those which can produce a reproducible, validated result, which cannot compromise with the safety, and therefore they can be accepted. And that is the strategy is establish internationally approved in vitro alternative methods in vivo model. You do it in India. And the second strategy is develop new and more sensitive cell-based model. And this cell-based model again is a very, very tricky thing, because we have to have a validation process, because if the oxygen concentration changes, if the pH changes, if the atmosphere, if the humidity changes, the results of this mortality of the cell change. And that's why many times when we say there's this cell-based assays are very difficult, it's very easy, but it is difficult because controlling all those external influencers is important. GLP compliant non-clinical safety tools are regulatory submission. All regulatory submission is today requires the animal experiment. And that's why just 2 days back, and when we thought that yes, we are sending a document to the government, I primed the entire committee, and then a list is being prepared that these are the potential areas where GLP will consider from as and when the evidence comes. Now, steps involved in developing alternate models. I think this is will be covered in different sessions. I believe the development of alternate model is the first thing. Publication is the first thing. That is the beginning of the journey. But important thing is pre-validation. In the lab itself, you validate with the control system. Validation means you have to do it at least 2-3 labs. Don't just restrict to publication. Do it validation and then pre-reviewed, and then ultimately acceptance by the regulator. Once this becomes, then we are successful. OECD guidelines, I think I was just talking just 3 days back in that GLP meeting. I was saying, everybody was saying, sir, OECD में लिखा हुआ है ये इन विट्रो टेस्ट, इसको हम कर देते हैं. ये OECD में लिखा हुआ है. अरे भाई, OECD हमारे खुदा तो नहीं है ना. They are not our God. Why not a time will come when we say that this is because this is introduced in GLP India, and therefore OECD will accept it, adopt it. And then when I found it, I talked to them. He said, if there is any model which in India you have validated, you have verified, then OECD will not be hesitant in adopting it, provided it is again valid. So now when the reverse, now India is becoming Vishwa Guru, we say. Now here is the product. The only need is that we have to market it once it's validated. The OECD will accept it. So I will request all of you that don't bhajofy, don't ratify every city, every time OECD, OECD. OECD is important in today's context, but maybe after 5 years, 10 years, you say it is IECD, Indian uh, guidelines, because of you have developed some of them. That is important. So I think these are the something which are already approved in OECD and we have to do it. Adverse outcome pathway approach, and this is something which is important. We will not go into the detail, but these are the process when you see what could be the possibility of predicting the outcome adverse safety concerns by alternate angles. Now alternate models yet to be developed, and that's why the organ and chips are organized may sometimes give an answer. Reproductive toxicity, carcinogenicity, reproductive toxicokinetic study, as on date, nobody has developed. But when you say there are models where you inject a drug or you inject a chemical which is poison in nature, you can predict by artificial intelligence, by some experiments. You don't have to have 1 hour, 2 hour, 15 minutes, 30 minutes, 1 hour, 2 hour, 4 hour, 12 hour, 24 hours, and then 1 day, 3 days, 7 days for normal pharmacokinetic structural science. You can do intermittent sampling or just one or two animals, and then you just do the algorithm by artificial intelligence, and you can predict with 80 to 90% accuracy the kinetics of the at higher dose, toxic dose, as well as kinetics in the dose. So that is what the models are required. The policy framework: alternate animals validated and accepted, alternate models validated but yet to be accepted, alternate models being developed, and lastly, alternate models. So, yes. Oh, sorry, my fault. That is it. Not understanding the technology correctly, but jumping on using it. I did not know that I pressed the button off. So, so abrupt ban or phase wise restriction. Maybe Peter can give it thought to it. We can give it thought to it. And are we alternate? So, so now this is why we say you identify the critical areas for and develop that. And what happens to the consumer safety and what about views from other stakeholders? The last statement is perhaps the key of success. It's not that we work on silos, we have a discussion here. We work, we debate involving different stakeholders, including those who are totally against our society, including those who are the policy makers, those who are regulators, to say all this is useless because you don't have enough. That is where we have to work. So need to develop a roadmap, and roadmap is academia, the society, the industry, and I'm happy to see here the regulatory people and funding agents. It's very difficult to get a funding for even this conference. What to talk of funding for Fortunately, Indian Council of Medical Research framed a committee on alternate to animals. And I was just talking to Dr. Raisuddin. This was just about to be released last year, but then the secretary changed, new secretary came. The entire thing went into, I would not say garbage, but into a pending box. Now it is being again revised, and maybe you may see in few months it will come. But this is where we have to put them again and again. In Indian perspective, I will not go into the detail experimental models. Lot of work has been done, but I would say zebrafish. If you can just see this, if you just see the website of the institution, CDRI, ITRC, IITs, our national GLP technical committees under which several CROs, they have been working on these in silico models, and then the different models, more than 20 to 30 models have been developed for different groups. And C. elegans and zebrafish has become a fashion now. If any lab is working zebra, why? Because it's simple. This is not a, maybe sometimes we can say zebrafish syndrome. Because everybody, but this is such a versatile model. You can do any type of, but then it has to be validated. And this is the one model where inter-laboratory validation can be done easily. And then you can see what it can do. C. elegans has been used for many, many years. In vitro, now this is a 3D organoids, and couple of IITs and JRF is also doing developing 3D. And Department of Science and Technology has funded a huge funding for developing 3D organoids. And organoids have been developed in IIT Chennai and IIT Delhi and also IIT Mumbai. Now, if once this is developed, then it is not the tissue on culture, but it is the entire system. And maybe at a later date, the clinical trial of the toxicity on the liver toxicity, the kidney toxicity, the brain can be assessed by 3D organoids. And that's what we are looking for. And that's what we call as a human on chip. And the human on chips has a lot of issues, but that may be in future, and that may require that yes, this has completely replaced animal experimentation, but then it requires much more understanding. So this is again 3D model, and this is the biodefense research. And now they say this is the, not only in India, globally this is being tried, and many times when we say that these type of research are for rich countries. अरे भाई रिच कंट्री का तो ठीक है, बट हमारी कंट्री में भी तो इतने सारे डेवलपमेंट हुए जो हमने दिखाए पहले. And that means it is not that it requires a very, very high tech, and we are not less high tech. If you just see the institutions of a CSIR like that, 3D culture, and then so this Indian initiative in 2022, they made agreements. See, when today in I chair the National GLP Technical Committee, and in 2010-11, first time the India become mutual data agreement, MAD. This is not mad. It's not mentally mad, but this is a pride movement that India became a mutual acceptance of data. Now, whatever data we produce in our GLP certified lab is acceptable. And today, it is of course by rotation like G20. The vice chair of OECD is DST India. And next year, by virtue of rotation, it will be chair. So this is the right time when we can influence. And of course, in 2022, this was reviewed 10 years, and India came out very flying color. This is ITA, and this is also the facility creation, 3D printing. The facility has been created in IITs by Government of India, and this is what the vision and mission of Dr. Akbar Shah and the society is to create the Indian Center for Indian Center. Remember, Indian Center for Validation of Alternate Models. And I had a just 3 days back discussion, informal discussion, when the Secretary DST came for conclusion remark of our GLP committee. He said, yes, you put up the proposal. So we will discuss this set and we will shorten it. I was just discussing with Dr. Raisuddin. So this is to see the primarily the objective of if this is set up, wherever set up, how it is set up, we do not know. But to do the validation process, to do the cross-working, to do the networking, to do the convincing to the regulators, and of course, to that. That is what the proposed objective of. So the way forward, developing a novel alternative to animal models, acceptance of the regulatory body. That's again, and quarterly symposium to update the community. This is what it has been proposed, but today it is. See, pre-corona it was not possible, but now it is possible because you can have the online meeting. The risk of online meeting is always, isn't it, Ankita? The risk of online meeting, the biggest risk of online meeting is that I put my switch on, I show my face, and then I do my all domestic work or maybe whatever it is. And at the end of the meeting, I again, if required, I will show my face. The rest of the time, I must not. So these are the risks. But then there can be methods. You ask question in between. Where are you, sir? So so if he is not there, if he doesn't respond, he is not there. The answer would be my mute. It was mute. It was not working. But all those are fraud statements. The only statement is that you were not there. That is the simplest. And that's why when it is linked with the, I tell you just, and I just finished, I tell you one simple thing. When Government of India observed that the several meetings on online, people just attend in the beginning, and sometimes not even the beginning, they just put it on. And at the end, hardly 1 or 2% are left because nobody speaks. What is the solution? Take photograph at the beginning, at the end, and if required, in between also, and link the remuneration with your presence. The moment the remuneration was linked, the attendance increased. So the remuneration 5,000, 6,000 rupees, if you are just attending, but you are not going, you will otherwise it. Now that is, so some checks and balances can be developed. But what I am trying to say here is we can have some brainstorming in between from our society. And I think the way forward, harmonization, regulatory flexibility, and all those things. So that is uh, just see. Okay. Sometimes when the time is off, these slides don't move. Anyway, the last slide was a river. A river means is the alternative, the established, and the alternate. It's very fast. If you have traveled, you must have seen Haridwar. And Haridwar, Har Ki Pauri, the flow of the water is so difficult to cross, and the temperature also. But what is the way? Can you cross it? Can you cross it? Can you cross? How many of you can cross it? Because this river flow is so fast, you can't stand. The temperature is so freezing, but can you cross? How many you can cross? I can do. Because I know that there is a bridge, isn't it? So we can create a bridge here, and that's what the alternative. Thank you very much.
Thank you, sir. Thank you for this enlightening session. I would like to request now Professor S. I. Sudin, sir, to present our speaker, Professor Y. K. Gupta, with a token of appreciation. Sir, please come forward. Thank you, sir.
Thank you, sir. Now I would like to invite Mr. Puran Chand, sir, to hand over the memento to Professor S. Raisuddin, sir.
Sir, the memento is mine. Chairperson to have few words on this session. Or any questions?
Thank you, sir. Now I request Professor Y.K. Gupta, sir, to honor Mr. Puranjan, sir, with a token of appreciation.
A few words from chairperson or some questions would be appreciated.
So, so I think it was a trend-setting lecture by our president, Professor Y.K. Gupta, and he has covered all the aspects of difficulties as well as opportunities, whatever SWOT analysis he has done. There are lots of opportunities also in alternatives. He narrated a story of frog that time, and everybody can relate that we used to sometimes enjoy suffering of frog, and we will put in the dalda container and put a chloroform, and when it will just struggle for his life or before he gets drowsed. And but now I think a lot of change has occurred in our Indian scenario. Very difficult to get approval for animals. Although sometimes we argue, we think that unnecessarily they are policing our research. But at the same time, one thing definitely has happened, that we are looking for alternatives. And I was looking for last 2 years. 60% research papers from university departments are now in silico toxicology, molecular docking, things like that, or the networking of the proteins, etc., etc. So India is also catching up, and I can see that I was seeing some publication for a university. I don't want to name. Almost all publications are in silico, and they are getting grants. They are also getting publications and PhDs. So definitely, it has been like this, and we are witnessing. And in this society, particularly, as I mentioned by Professor Gupta, is मैं 2 मिनट बोलना चाहूँगा. Then it is growing. We are happy with the limited number of membership. It's, I think, 70 only. But definitely, I would urge the young people and other to join. At least from Aligarh, there should be a campaign for membership for this society, and we will definitely bring qualitative as well as quantitative change in thinking of. And already mentioned by Professor Y.K. Gupta, ICMR and DST is very keen to about this validation aspect. So Ankita and Professor Akbasha and other people will definitely come up. Since it was a talk which is a keynote or leading talk, generally there is no question on that. But still, Puran Chand ji साहब कुछ आप कहना चाहिए? Please, you say few words, whatever you नहीं, आप कुछ अपना पर्सपेक्टिव रेगुलेटर आप हैं. So I think very important role regulators will play, drug inspectors and drug regulators and SEDESCO. तो ये दिल्ली डिवीजन के हमारे वो हैं पूरन चंद जी, रेगुलेटरी अफेयर्स को देखते हैं. तो आप अगर कुछ हिंदी इंग्लिश जिसमें भी आप मुनासिब समझे यू कैन.
सभी को नमस्कार. मैं सभी लोगों का इस अलीगढ़ की पावन धरती पर पहले तो स्वागत करना चाहता हूं मैं, और बस इतना ही कहना चाहता हूं कि जो टॉपिक है वाकई बहुत ही विचारणीय है, और कि क्या अल्टरनेट हो सकते हैं, बहुत गहरा है इसको देखा जाए तो. और मैं उस चीज को एनिमल क्रूरल्टी से भी जोड़कर देख रहा हूँ इस चीज को, कि किस प्रकार इसको मिनिमाइज किया जा सकता है, किस प्रकार हम जो एनिमल के साथ होने वाली जो चीजें हैं उसको किस प्रकार और अल्टरनेट क्या ढूंढ सकते हैं. बस मैं इतना ही कहना चाहूंगा. यहाँ काफी 2 दिन का सेशन चलेगा और काफी विद्वान लोग आए हुए हैं, और मैं धन्यवाद दूंगा ऑर्गेनाइजिंग सेक्रेटरी रहमान जी को कि इस मंच पे मुझे बुलाया. और बस इतना ही कहना चाहता हूँ कि 2 दिनों में कुछ न कुछ समाधान निकालिएगा कि देश दुनिया के अंदर कुछ न कुछ चेंज हो सकता है. कि अपने अंदर भी एक दिल है छोटा सा, वो भी कहता होगा कहीं ना कहीं किसी बोर्ड पर आकर कि हम क्या कर रहे हैं। ठीक है, मजबूरी है कि हम लोगों की कि एक दूसरा पहलू देखते हैं कि कहीं हमें कुछ रिसर्च करनी है और उसमें कुछ कामयाबी भी हासिल करनी है। तो मैं इतना ही कहना चाहूँगा कि धन्यवाद आप लोगों को और कि मुझे यहाँ आमंत्रित किया, और इन 2 दिनों के अंदर कुछ ना कुछ निकलेगा, निश्चित रूप से निकलेगा कोई ना कोई रास्ता। और एक सौभाग्य है डॉक्टर वाई के गुप्ता जी का, मैं काफी नाम सुना था और आज सौभाग्य होगा कि मेरा आज रूबरू इनसे मिलने के जो मौका मिला। नमस्कार।
Once again, thank you all our esteemed guests for this wonderful session and sharing your wisdom. For our next session, I invite Dr. Surabhi and Dr. Pragya to take over the podium.
Good morning, ladies and gentlemen, esteemed guests and delegates. I am Dr. Pragya Roy,
And I am Dr. Surabhi Jain.
Junior Resident from Department of Pharmacology, JNMC AMU. This session is a presentation by the winner of the Dipti M. Kapoor Endowment Award. It is my honor to introduce Dr. Gopinath Pakirisamy, a luminary in the field of biotechnology. Currently serving as a professor at the Department of Biosciences and Bioengineering and heading the Center of Nanotechnology at IIT Roorkee. Sir's academic journey began with a BSc in Microbiology and an MSc in Biotechnology from Hardi Desan University. His pursuit of knowledge led him to earn a PhD degree in biotechnology from IIT Guwahati, followed by impactful postdoctoral research at the University of Rochester Medical Center in New York, USA.
A true innovator, Sir holds a remarkable 19 patents, both granted and filed. In addition to his prolific research, he has lent his expertise to numerous books and chapters. Recognized for his outstanding contribution, Sir has been the recipient of numerous awards. Without further ado, let us welcome Dr. Gopinath Pakirisamy to the stage to enlighten us.
Yeah, thank you very much for the nice introduction, and I'm happy to be here in this one of the oldest institution. And again, I'm from another oldest institution, IIT Roorkee. So last year we celebrated our 175 years of our institute. So, can anyone help in that? Not okay. Not okay. Yeah. So first of all, I thank the jury members and award committee members for selecting me for this prestigious award. And the title of my talk is 4D scaffolds, adding a new dimension to tissue engineering. So in my talk, I'll broadly cover about what is tissue engineering and what is the role of nanotechnology in tissue engineering. And I'll explain about what is 4D scaffold. So before we go into the topic, let us see a brief statistics. So if you see the need for organs today in US alone, approximately 1,19,000 people require organ transplantation. And out of that, only 30,000 is able to get the organ transplantation. And every day, at least 22 people are dying due to lack of suitable organ. So then you can think about the whole world, how many people are losing their life due to lack of suitable organ transportation. And if you see then India statistics, approximately 1.5 lakh people need a kidney transportation, but out of that only 3,000 are able to get it. And this is the statistics. And problem is, like, if I ask you the cost of your mobile phone, or if I ask you the cost of your car or bike, you can easily tell. But if I ask you the cost of the organ, you cannot tell. What is the cost of your lungs? What is the cost of your heart? You cannot tell. According to this website, this is the cost. Again, it is not the legal approved price or whatever it is. According to this website, this is the cost of each and every organ. So we don't know the value of organ, and if it is not functional, then we'll realize what is the real value of the organ. So while teaching to my students also, I always say that don't ask any property from your parents. They already given this much worth property to you. And also, if your parent says that you are useless or worthless, you can show this slide. You can show that how much worth you are, right? So the idea is, what is tissue engineering? It is a multidisciplinary field where you need the help of biological engineer, chemical engineer, metallurgy, and electrical engineer. So what is the main motivation for this tissue engineering is, suppose if there is any organ damage or repair, so we can replace the diseased tissue or damaged tissue by making the artificial tissue in the lab. That is the idea of tissue engineering. And this is the overview of tissue engineering. In tissue engineering, what we'll do, we take the stem cells from the patient body and we can grow in the lab 2D environment. Then we can put it in the 3D environment. Then once you grow that cells or the tissue, then we can re-implant into the patient body. So in this case, the rejection rate will be less or nil. So that is the idea. And why three-dimensional? Why not 2D? So usually we grow the cells in the lab like this kind of two-dimensional glass plates, right? But that won't mimic your original body organ. Inside our body, we have all the organs in 3D, three-dimensional. So to mimic, we need a three-dimensional environment. And these are the three important things required for the tissue engineering: the cell source, and ECM is extracellular matrix. So the cell source may be a kind of adult stem cells or embryonic stem cells. Again, embryonic stem cells in most of the country it is banned, because to get the embryonic stem cell you need the aborted fetus. And adult stem cells, no problem, you can take it from the patient body, the adult. And the scaffold, so this is the extracellular matrix. Depends on the organ, the scaffold will vary. If you want to replace the hip joint or knee joint, we have to use the metal scaffold. If it is bone, you can use the ceramic or polymers. And third important thing is the signals. So the stem cells don't know whether it has to become like liver cells or kidney cells. So these growth factors and drugs, which is going to decide. And again, one of the important parameters which I'm going to explain, that is the mechanical forces. So this mechanical force also plays a very, very important role. So for example, you might have also observed the students or the boys stays in the same hostel eating the same mess food, but the person who goes to gym, he get very good muscular development. So what is the reason? So when you give the mechanical stimuli, so that will induce the gene expression, that will induce the protein expression. So that is called mechanotransduction. So we are also developed a scaffold based on this mechanical force, which I'll explain more in detail later. So what is the role of nanotechnology in the tissue engineering? So mainly we'll be focusing on this scaffold. So initially scientists started with the micropore scaffold and microfiber scaffold. If you see that the size of the cell is also micrometer dimension, so it is attaching only on the fiber axis. So it is not mimicking like a original tissue. So for that, we have to go for nanofiber. So when you go for this nanofiber, so the scaffold, the foundation is in the range of nanometer and the cell is in the range of micrometer. So by using this nanofiber, we can closely mimic like your original extracellular matrix. Then we can make the artificial tissue. So you can see here, this is a single cell, and single cell is on the thousands of nanofibers. So if you make such kind of things, that will mimic like your original tissue, and the signals can be easily transmitted. That is the one important thing. So what is nanofiber? So just to give a broad idea, so this brown color is the human hair. So the human hair is 80,000 nanometer, and you can see these are these nanofibers. So then you can understand how much small is these nanofibers. So these nanofibers have a lot of application. in our lab also we are working on all the applications except this the bulletproof jackets. Almost all the applications in our lab also we are working. How to make these nanofibers? There are 3 methods to make these nanofibers: self-assembly, phase separation, and electrospinning. So out of that, this electrospinning is one of the simple method. Using that, you can make these nanofibers. And what is the principle of this electrospinning? And the electrospinning, when you apply the high voltage, then it will start making the fibers. So the principle is similar to this cotton candy. So most of you might have seen this in your school days or in the exhibition. So what you do, you add this sugar. So when you add the sugar crystal here, so the sugar will melt, and sugar is a polymer. So, and due to this high electric voltage, it makes the fibers. So basically what we are eating is microfiber in the cotton candy. So if you control it in a better way, we can have these nanofibers. So instead of putting the sugar, we can put the FDA approved biopolymer. And instead of this pink color, you can put our therapeutic material, then apply the high voltage, then you get these nanofibers. So these nanofibers can be collected in the grounded collector. And again, it can be a stationary collector or it can be a rotating drum collector. So when you use this rotating drum collector, so you get the aligned fibers. So you can see here, when you use the stationary collector, you get this kind of random fibers. And if you're using this rotating collector, it will be like a kind of thread bundle. It will have the aligned nanofibers. And once you make this fiber mat, you have to check it whether this is hydrophilic in nature or not. So how do you check it? You can just simply put a drop of water and take a photograph and measure the contact angle. So if the contact angle is less than 90, that means it is hydrophilic. If it is more than 120 or 150, it is hydrophobic. So the simple example to understand is just put a drop of water on the plastic sheet. It won't absorb, so you'll get this super hydrophobic. And you put a drop of water on the wooden surface or any surface, it will take up the water. So it is hydrophilic in nature. So let us move on to this, the main topic, what is the 4-dimensional tissue engineering scaffold. So here, what we did is, basically most of the lab, they are working on developing the 3-dimensional scaffold. Because the 2-dimensional, what we are using in the lab, which cannot mimic like your original tissue or original organ. And alternate is you have to go for animal trials. So as Sir suggested, Professor Vaike Gupta, so we have to reduce these animal models because it has a lot of cost, time consuming and ethical issues. What we can do is we can go for 3D scaffold. Most of the lab, they are going for 3D scaffold. But the problem is the 3D scaffold, still it is not able to mimic like your original organ or original tissue. So for example, so the 3D scaffold is static, but inside our body, the organs are dynamic. For example, you want to make an artificial heart or artificial lung, so the scaffold should be able to shrink and expand. So to overcome that challenge, we made this 3D scaffold, and in addition to that, we added the magnetic nanoparticle on the sides. So when you have this magnetic nanoparticle on the sides, and by using this external magnetic field, you can make the scaffold to shrink and expand. So that is the idea. So by using this external magnetic field, we can make this scaffold to shrink and expand. So it can mimic like your artificial heart or artificial lung. Those kind of things can be simulated in the lab. So you can see here after this giving this mechanical stimuli, so all the cells are organized in a particular orientation. And again, it is similar to the Lego bricks. When you use the Lego bricks, you can make any kind of structure. Inside our cell, we have this small subunit that is called as G-actin. And this polymer is called F-actin. So based on your external stimuli, it can be a nutrient source or hormones or growth factor, or it can be a mechanical stimuli. So what happens is this G-actin, it will assemble and forms like a final polymer, that is a polymactin. Okay. So when you have this, yeah. So the base depends on the mechanical stimuli, it depends on the external stimuli. So it can be the G-actin become a F-actin. And based on the mechanical stress or mechanical stimuli, so the cells will extend this kind of stress fibers. So you can see here, with respect to the mechanical stimuli, this actin and vinculin, so these are overexpressed with respect to the number of cycles. So if you do the mechanical stimuli for 3 cycles, you can see the expression of the cytoskeletal protein like vinculin and actin is overexpressed. And when you give the more stress, then these cells will have like this kind of filopodium, that is like a finger-like structure. When you stimulate the cells, it will have like finger-like structures that is called filopodium, and that can be studied by using this mechanical stimuli. So when you use the mechanical or magnetic stimuli, you can see this is a single cell, and these are the nanofibers. So the simple analogy is when you go in the roller coaster, we hold our sister or mother hands, and nowadays it is like a girlfriend or boyfriend. So we hold it, and here you can see here this single cell, it is grabbing this nanofiber with respect to the mechanical stimuli, and the displacement is approximately 5 to 10 micrometer with respect to the mechanical stimuli. And this scaffold and this kind of device have a lot of applications. And if you see that currently the three-dimensional scaffold have a very huge market. Most of the lab, they're trying to make this kind of three-dimensional scaffold to reduce the use of animals. And again, our work is slightly different. So when compared to 3D, 3D is a static. In our cases, it can be dynamic. We can make the scaffold to shrink and expand. And also this is low cost when compared to the commercially available things. And again, this can be kept it in the any microscope, and we can use it for imaging applications. So if you see the cost of this three-dimensional scaffold, this is very huge, the branded one, and ours is 100 times or 1,000 times cheaper than this. And if you want to make this artificial tissues like artificial heart or artificial lungs, if you want to repair that, and those kind of stimuli can be given in the artificial lab environment. So when you transplant these tissues to repair the heart or lungs, and it can match with the particular mechanical load, and it can easily heal the wound. And not only that, the stem cells can also be differentiated. So for example, the stem cells don't know whether it has to become like a liver cell or kidney cell, and for that we have to add the growth factors or hormones. But instead of adding the growth factors and hormones, can we give this mechanical stimuli and make the stem cells to differentiate and proliferate? So that can be also achieved by using this kind of device. And this device can easily go into the CO2 incubator, and also it can be kept under the microscope also. It is transparent. So we can easily see that cells growing, and we can do the live cell imaging also. So for this work, we filed a patent, and it has got done a technology transfer also to a company. And also this was selected for the cover page of the issue. And we got the prestigious Gandhian Young Technology Innovation Award from the Vice President of India. And this, based on that, they gave a 15 lakhs fund to develop this into final product. And we also developed the product into commercial stage. So not only the mechanical stimuli, now we can do the electrical stimuli also. So when you do the electrical stimuli, we can make these nerve cells, and you can understand how these nerve cells can be regenerated, how these nerve cells can be communicated. So all these things can be achieved. So due to IPR issue, I'm not showing those slides. And not only the 4D scaffold, based on the nanofibers, we are also making some other products also. So the first one is nanofiber based anti-cancer drug delivery. So here we made this core shell nanofibers. In the nanofibers also we can make a core and shell, like a pen and refill. So the advantage is you can load one type of anti-cancer drug in the core, and in the shell, the red color region, you can load another type of anti-cancer drug. So if we have two types of anti-cancer drug, the efficiency can be improved. And this can be mainly used for the post-surgical. So whenever you remove the tumor, the problem is the tumor will reoccur if the doctor not removing the complete tumor. In this case, you can put this kind of anti-cancer drug-loaded nanofiber, and you can see here this thread-like structure. These are nanofibers. On the top of that, you are having these cells. So when the anti-cancer drug is released, the cell is dying. So that's why the cell become the long cell. It become like rounded. So that means it is able to kill the cells. It will prevent the reoccurrence of tumor. So after the surgery, this bio bandage can be placed, and it can act as a physical barrier, and also it acts as a kind of slow and sustained drug delivery system. And we also made this bio bandage for the mainly for the diabetic. So in the diabetic wound, the problem is when you put the normal bandage and when you remove the bandage, the wound get disturbed. So here we made the bandage bio bandage. It will be biodegradable. So you can put it on the wound, and it will heal the wound faster. At the same time, it will degrade in the wound itself. So that is the additional advantage of this bio bandage. And another thing is, whenever we want to study the wound healing, So we have to depend on these imported items like this corning and the cell culture insert. And the cell culture insert costing from 500 rupees to 5,000 rupees. So in our lab, we made a very simple one. We taken these micro tips and we cut the backside and we deposited the nanofiber on the top of that. And the good thing is you can do autoclave, you can do sterilization because the silk is, it can withstand temperature even 120 degrees Celsius. And this can be useful for understanding these cell mobility, wound healing studies, how the cells move. So those kind of things can be studied using this kind of simple cell culture insert. And the cost is around like 1 rupee or 2 rupee when compared to this 500 or 5,000 rupees cell culture insert. And not only biomedical, we also done something relevant to environmental. For example, this is a water filtration setup like fiber. So when we use this RO filter, there are a lot of waters are going waste. And this is like a kind of membrane. So which we use in our village site. We use a simple cloth and filter the water. So this is like a simple nanofiber based cloth. And this is a bacteria. You can see this is a 10 power 6, 10 lakh bacteria. And we use this green fluorescent bacteria. And you can see this water is filtered and there is no green fluorescent. This is completely the clean water. So that is also possible. And not only that, it can also remove the heavy metals. So mostly like in the Calcutta and other regions, there are heavy metals are mostly in the water. So those can be absorbed on this membrane. And during COVID time, we also done the mask, the normal 2 rupees mask. It has the middle region open. So in the middle region, we can put our nanofiber and we can improve the efficiency of this 2 rupees mask to equivalent to N95 mask, which is expensive. And again, it is a kind of, you can throw this mask in the dustbin also. This mask can also remove the bacteria and other things. So that is the additional advantage of this mask. It's an eco-friendly mask. So the final last slide is like not only for the, again, environmental, we can also use for the food. So nowadays, if you see this apple and other fruits comes with the wax coating, which is a potential carcinogen. And so to overcome that, so we use a silk fibroin, that is a biomaterial. And we also use this curcumin, and which is having this antimicrobial and antioxidant property. And we use the honey, that is a moisture absorber. And when we use the banana as a model system, and you can see this, it is able to stay even after 10 days also, the banana is fresh. So some of the company also approaching us for spraying on, because when they export this banana, it gets spoiled in the shipping. So we can also use it for, it's a kind of edible packaging. Instead of using this plastic packaging, which is again environmental waste, again the fish and other things will eat and use a problem. So this eco-friendly and environmental package. So with this slide, I'll conclude my talk, and I thank all my students as well as the funding agency. And this is my research group, the present research group. And I welcome you all to our IIT Roorkee. This is one of the oldest institute in the country. So last year we celebrated our 175 years, and this is our 175 years old building. So I'll happy to invite you whenever you get a chance to visit Roorkee. You can visit our lab, and if you have any questions, I'll be happy to answer. You can use this. And mine. So I have got to do this.
Selected for this award. Dr. Gopinath Pakirisamy has been selected for this award, and he has made the presentation. And he presented to you the kind of discoveries he made, which made him eligible or competent to receive this award. I, as the General Secretary of the Society, have to tell you in a few lines about this award, what this award is. Well, the Society for Alternatives to Animal Experiments was established in 2018, about which I shall have a lot of things to tell in the afternoon. And From that time onwards, we have been very elaborately supported by two organizations, NGO organizations, the People for Ethical Treatment of Animals, PETA, and Humane Society International, and not to mention the People for Animals. Now, at that time, we had one young scientist, very active, very dynamic, associated with the People for Ethical Treatment, who have been very strongly supporting us. And the strong support to PETA was essentially in view of that person. The person was associated with the PETA as a scientist, the science policy advisor, and her name is Dr. Ankita Pandey. Very nice person, very nice, NGO organization activist, and also a scientist, a serious scientist. So she was a very strong support to the society, and unfortunately, I'm sorry. Dipti, I'm sorry, really very sorry. Dipti Kapoor. Ankita is so. So Dipti Kapoor was the person who was at that time associated, and she passed away at a very young age. And so we, the society people, thought it's very fit to have an endowment in her name to award a prize to a person who has worked on alternatives. So that way, the thought was spread among the members of the society, and we were able to mobilize some supporting grants, and sponsorships have come from different people, and we equally good amount came from PETA, from members and the PETA. And the idea was that the money will be deposited in a fixed deposit, and the proceeds will be awarded as a cash prize to a person who has done good work on scheme of alternatives during that particular year. So we launched it in 2021, and we had the first award presented at 2022 at the conference at Thiruvananthapuram, the 5th annual conference. And the awardee was Dr. Shilpi Sarma, who came from University of Pune, Department of Biotechnology. And this is the second year, and we invited nominations. We received 6 nominations, and all the 6 people are competent enough to be considered for the award. Very good scientists, all from institutes, universities and institutes. And we nominated a very highly efficient juries, at 3-member committee, and the jury considered the proposals that came in all possible seriousness, and the committee recommended the award to be given to Dr. Gopinath Pakirsamy, who comes from IIT Roorkee. That doesn't mean that the other 5 contestants are not doing good work at all. They are close to. They are in fact neck to neck tie, and ultimately the final decision went in favor of Dr. Gopinath. And you should have been convinced by now that based on the kind of research output, contribution to the alternatives movement through not only 3D culture, but more than that, the next level of cell culture, which is not much spoken about, the 4D culture, which is actually a system, a mechanism by which you can develop the test material, which is human rather than animal. This is how the link goes. His research will be connected to development of model testing systems, which will be human rather than from animal systems. So his discovery takes this test more close to the human system. So there's human stem cells are cultured into organoids, and the organoids will be used for testing toxicants and testing pharmaceuticals. So just to tell you what this award is, and how Dr. Gopinath gets himself declared for this award. Thank you for listening to it. I thought you should be told about what this award is. That's why I came in. Thank you very much.
Thank you so much. The award ceremony will be done during the inaugural program. Thank you so much.
The award will be given during the inaugural session.
Thank you, sir, for your amazing presentation.
Now I am pleased to hand over the podium to Dr. Anam and Dr. Abhinav for the next session.
Assalamu alaikum and very good morning to all. I, Dr. Anam, Junior Resident.
I, Dr. Abhinav Tiwari, Junior Resident.
from Department of Pharmacology, JN Medical College, AMU. We would like to invite our chairperson, Professor Dr. M.A. Akbar Shah and Dr. Adip Roy sir to come over the stage and chair the session. Though our worthy chairpersons are in no need of introduction, as their achievement in the field of science resonates throughout the world. Professor Dr. M.A. Akbar Shah sir is currently the General Secretary of the Society for Alternatives to Animal Experiment and Editor-in-Chief of the Journal of Endocrinology and Reproduction. He had been the director and chair of the National Center for Alternatives to Animal Experiment under the University Grant Commission. Sir is identified as the Gandhi Grover Durand Camp Chair for Alternatives in Education and in Vitro Toxicology. Sir has a very distinguished career as a scientist and a teacher for 38 years. His area of research includes endocrinology, reproductive biology, cancer, obesity, in vivo and in vitro toxicology, and alternatives to animal experiments. Sir has conducted more than 100 training workshops on alternatives to animal experiments with experts from India as well as abroad, and popularized alternatives among academia, scientists, and the regulatory community. Thank you.
Dr. Adip Roy, Associate Vice President of Regulatory Affairs in Amway India, is a senior regulatory and toxicology scientist with expertise in chemical safety risk assessments and natural safety risk assessment for use in consumer products. He has contributed to the national agenda on risk-based approaches for safety assessment of cosmetics and food products and is a leading contributor to strategies on alternatives on animal testing in India. In the field of research, he has over 8 years of experience in neuroscience, especially in addiction biology and learning and memory. Now I request Professor Dr. M.A. Akbar Shah and Dr. Adip Roy to chair the session and introduce Savita Nutal for this session.
Thank you very much, everybody. And it's really heartening to see such a conference. And thanks, Dr. Zia, for organizing this. See a lot of old friends and colleagues over here. Really, it's really pleasing. And and just before the conference, I really had the privilege to speak with Dr. Savita. And looking at the two presentations that has happened, a session by Professor Y.K. Gupta and Professor Gopinath, how much science has progressed. I mean, previously we were just looking at replacing animals one to one, and now looking at the progress in science. Dr. Gopinath talked about biopolymers, the 4D cultures, and on that context, let me just introduce Dr. Savita to you. I mean, she is currently a lecturer teaching health and social care management at Fairfield School of Business, is a former NHS clinical scientist and a passionate advocate for animal-free medical research. Over the past decade, she has studied, trained, and worked with medical sciences sector and has first-hand experience in how potential medicine tested on animals has kept medical research from moving forward. She has created Medicine Without Cruelty in January 2023, an educational organization dedicated to promoting medical research without animal cruelty and seeks to educate and explore animal-free innovative technologies. When I talked to you in the morning, you were talking about genetic research challenges and its utility in the outcome of animal experimentation. We are just looking forward to hear from you. Thank you very much, and the stage is all yours.
Thank you so much for the warm introduction, and thank you for inviting me here today. I'm very grateful to Dr. Akbar Shah and Dr. Rahman as well for the warm invite. No. No. Oh, there it is. Okay, thank you very much. Okay, great. To quote the esteemed philosopher Ralph Waldo Emerson, there are always two parties, the party of the past and the party of the future, the establishment and the movement. Throughout the early part of my career, I was deeply immersed in understanding the establishment's practices, and I was deeply conflicted with the research that was happening, the animal testing specifically. Today, I speak before you. I'm not just here as a speaker, but as an advocate, wanting you to join in what I can only believe is not merely a cause, but a movement, the animal-free movement, where ethical science and respect for all forms of life are not just flowery ideals, but they are clear, feasible realities. And so I want to share with you my path to enlightenment, so you can call it, my embarking on the path to truth. I spent the last decade working within the field of diagnostic genetics and complex diseases, and why I began the journey called Medicine Without Cruelty. And finally, I want to discuss corporate practices from large companies and the deceit that's involved in all of it. So I want to start with my first truth. Genetic similarity does not equal physiological similarity. Now, if everyone must have heard of Darwin's theory of evolution, but animal researchers to date have used Darwin's theory to say that because we share a common ancestor with species and because of the genetic similarity with them, that is reason enough to justify using animals as proxy to humans. Well, I'm going to talk about how my career blew that idea to pieces. And I want to talk to you about rare disease. Now, if you don't know the world of rare disease, but there are actually at least 250 million people worldwide who live with a rare disease. That equates to about less than 1 in 2,000 people. And about 80% of them have a genetic component or a completely genetic basis of their disease. You might think that just because they are born with that genetic defect, as you could call it, that they would be diagnosed at birth, but that is far from the truth. The complex nature of the disease means that most will go undiagnosed for years, even for a lifetime for some of them. It might involve them going to multiple trips to the doctors, having multiple tests, having the wrong diagnosis applied to them at the beginning. And, it might even cause irreversible damage because by the time they found the cause of disease, it is far too late. Now, I believe ending this, what I like to call the diagnostic odyssey, this journey these patients undergo, has to be a two-pronged process. It is not only the sequencing capabilities that we have before us, the technology, it is also the human relevant evidence that we need to be able to end this diagnostic odyssey in order to serve our patients as best as possible. And so with the advent of whole genome sequencing, we can sequence the entire DNA of an individual in the matter of hours. It's incredible. And in England and the National Health Service, we provide that for patients with rare disease, fully funded. Now, what happens is the resulting DNA sequence we get from a patient is matched to a normal reference genome to identify any mismatches or genomic variants, as we like to call it. Those genomic variants undergo multiple levels of filtering. So we get rid of the common benign variants and we hone in on the gene variants of interest that are related to the disease. And sadly, animal models still remain a feature of functional evidence when we are classifying these variants. You need to remember, though, that the cornerstone of classification guidance is that the genetic change must fit the clinical phenotype, i.e., the gene change that we identify have to fit the clinical characteristics of the disease. And you cannot do that without human-specific evidence. And that's what I always advocated for. I always advocated for using, say, real functional evidence from the patient, bringing together multiple clinical specialities in the same room so that we can discuss these patients in a multidisciplinary team meeting, because it's so important to see what other pieces of evidence can be highly applicable to the patient, or see, for example, whether the genomic variant segregates with affected family members. And this is what we did in the pioneering program, the 100,000 Genomes Project. So that was the pilot program before we rolled out whole genome sequencing to the whole nation. We sequenced 100,000 patients and family members with rare disease and cancer and acquired cancers. So we were able to really kind of pioneer novel techniques, such as, what specific brain MRI scan images can we say closely relates, highly relates to the gene of interest. Now, replicating the phenotype, and a lot of people think that in order to examine complex phenomenon, you have to be able to simplify it, hence the use of models. That's what we're here for. But time and time again, we see that particularly for complex diseases, they are just not suitable. And I've just taken a snapshot of the different models that we have for complex diseases and why they don't work. Please do go ahead and look it up. I only want to talk about Parkinson's disease for now. We currently use rat models for the disease by destroying dopamine-producing neurons. But that method clearly does not replicate the progressive nature of Parkinson's disease. And recent cohort studies, such as the campaign study, it followed 142 patients from the UK over a period of 10 years, something you could never do with an animal model. And it demonstrated that clinical heterogeneity of the Parkinson's disease patient population. Now, your treatments are only going to be as effective as the models that you have to create it. And if you can cure Parkinson's in a rat, but that does not mean that you can cure Parkinson's in a human, because it clearly does not translate. And let's not forget that there are clear times when humans have suffered detrimental effects of animal testing. I've just, again, created a snapshot, but please do look it up. Follow the evidence. There are so many notable examples in which animal trials have directly caused devastating harm to human health. I want to touch on thalidomide and Vioxx. So thalidomide over here has always been a very controversial debate between animal researchers and anti-animal testers. It causes devastating limb malformations in an estimated up to 30,000 infants. So these malformations were caused by providing that thalidomide drug, which was marketed as anti-nausea medication for pregnant women. Now, it was safety tested in 10 strains of rats, 11 breeds of rabbit, 2 breeds of dog, hamsters, primates, cats, armadillos, guinea pigs, swine, ferrets. How many more animals are we going to test in it in order for us to realize that it's not working? After they found these kind of devastating teratogenic effects of thalidomide, they did test it in 3 to 4 different species of animals. So mice, rats, guinea pigs, pregnant animals, they tested in it and they saw no malformations whatsoever. So then what are we doing? You know, the cornerstone of scientific practice is we need to have a clear experimental design. Thalidomide shows us an appalling scientific practice. If you know your method is flawed, you don't repeat it again and again, so you can get that one fluke chance out of 100. Yet animal testers justify that thalidomide is kind of the poster child, as you like to call it, or propaganda, to prove that we need to have more rigorous animal testing. No, we need to throw out the experimental design in the first place and think about the in vitro tests that are already available, notably the embryonic stem cell tests. Now, Vioxx is another one. It's just here at the bottom. Now, that showed us the magnitude of how devastating human health can be affected. It caused 320,000 cases of heart attack, stroke, and heart failure worldwide, and was fatal in about 140,000 of them before being pulled in 4 of them in 2004. Now, they found in 6 animal studies in 4 different species that showed protective effects against heart disease. Why are we looking at this? Why are we using these models in the first place? We keep flogging the same idea over and over again, and we are not learning from our mistakes. Good scientific practice means owning your mistakes, showing clearly that it's not working, and how do we think outside the box without animals? Now, King and Wilson's hypothesis is almost 50 years old, but it's so key that variable gene regulation is the key to species differences. It's not the genetic similarity between us and them. It is gene regulation and how we can silence and express genes in our body, which makes us uniquely us. And I'd like to refer you to Dr. Jared Bailey's review that he did in 2011. Now, humans and non-human primates are a classic example because we are about 90 to 93%. I think chimpanzees are like 99% genetically similar to us. However, that divergence is those differences in gene environment is crucial, from chromosome structure to chromatin structure to post-translational modification. Genes can be expressed, silenced, regulated. And so this superficial similarity between human and monkey genetic sequences is of no benefit to biomedical research. Now, what was once unthinkable has finally occurred. We finally have a complete gapless sequence of the human genome. It's incredible. And that was only done a year ago. We addressed the remaining 8% of the genome, and we're missing the Y chromosome, which has also recently been sequenced as well. And this has culminated in 182 mega base pairs of sequence and we found that there were known errors in our previous genomic map. So the best way of explaining this is we have a better map of the entire DNA of a human. Isn't that amazing? And we found at least 4 new medically relevant genes. At least. We are still finding out what makes this T2T genome map. We're still finding out what genes are going to be implicated in human disease. And don't forget, there's also our non-coding genome as well. Now, this offers a stark reality to us. We are still unraveling the genome. We don't know what lies ahead of us, and we cannot be trusting animal models to truly replicate human diseases and complex disease processes. We know genes don't work in isolation, And we need to be clear that human relevant models are no longer a choice, but a medical necessity. I'd like to bring you to my next truth. All truth passes through 3 stages. First, it is absolutely ridiculed. Second, it is violently opposed. And third, it is accepted as being self-evident. Now, if you are ever talking to someone who does work in animal research and you're still stuck at stage 1 or stage 2, you should ask yourself, what do they stand to benefit from doing this animal research? And so I embarked on this new way of life, my company called Medicine Without Cruelty. It was founded this year, and it was to shine a light on the horrors of animal testing. And we need to be able to use clear language. Words have power, and the power to educate people should never be underestimated. If we are going to make a paradigm shift, we need to have clearly labeled words: barbaric, torturous, a mistake, devastating human health. We should learn from our historical, history, and learn and be able to promote animal replacements that are already out there. And so I provide online workshops, I write articles, and one of the latest was when I attended the World Congress on the alternatives to animal testing. And it was there that I learned about what I like to call the corporate deception. Large corporations and their heavy investment in cruelty-free PR. Now, remember, cruelty-free is not some sort of protected legislation. Just because it's cruelty-free does not mean that it is 100% cruelty-free unless it gets certification. And 2009 saw the implementation of the EU's ban on testing cosmetic components. However, there was another competing legislation which meant that single ingredients could still be tested, even if it was single use in cosmetics only. Now, in the UK, at least, the public was outraged because this ban was kind of essentially lifted for the past 25 years, and they were still testing on cosmetics. Now, that ban got reinstated. However, the issue is a lot of cosmetics have a dual purpose. They are not solely used in just cosmetic testing. They're used in other industries such as pharmaceutical research. And so even putting that ban back in place about cosmetic testing, unfortunately, that only covers about 20% of cosmetics, rendering that piece of legislation almost useless. Now, most companies may say they're cruelty-free, but if they are testing in countries where it is required to provide animal data, they are not cruelty-free. And many of these companies may say that they are working with, say, for example, mainland China, working with their legislation. The fact remains that they are still testing there. And this is done by third-party animal testing. So the blame is shifted. They are working with contract laboratories. So the testing that occurs that occurs on their products is not done by the brand itself, by the contract laboratories instead. So it could mean that the animal testing is done by an ingredient supplier, a lab formulating the products, any affiliate company, or a lab hired by the government agency. Animal testing can occur in a variety of combinations listed as well. And so that transparency is kind of gone between these large corporations and the level of animal testing that they do. And it's still not, it's not transparent because it's technically, they're not the ones who are doing the actual animal testing, but they are still paying for it to be done. So their hands are not clean. And it is yet again another smokescreen that we need to move past from. And if you look back through history, there's always been some form of scandal. When it was, say, Nestle, Unilever, Knorr, Hellmann's, and Yakult accused of animal testing, they were also accused of animal testing for the health benefits. So they weren't actually testing for the welfare of, human health. They were testing to identify whether there was some sort of health benefit, for example, in Lipton iced tea. I know this is an historical account, but I want to show you that this has moved forwards. So now what we see is, where we might have the big names, we still see that undercover investigations and exposés are showing cruel practices in these contract testing labs. And so we need to think about that term, cruelty-free. I mean, a lot of these companies have invested in heavy amounts of PR in showing that some of their products are cruelty-free by receiving, say, certification, for example, PETA certification or Cruelty-Free International. But that, again, another smokescreen. At what cost? They have a proportion of their brands that are cruelty-free tested, but the rest are still undergoing these third-party testing. Their hands are not clean. And it's time that we stood up and we recognized when there are animal cruelty practices still happening. Now, Charles River is a notable example, and it's someone that it's a contract laboratory that I identified at the World Congress. They have a 4 R's mission, so reduce, refine, replace. And what's the fourth one? Responsibility. Now, I'm offering a stark contrast to what you've probably heard before. I don't believe in the 3 R's at all. We could add a few more R's. It's repetitive, it's redundant, it's ridiculous. The more R's we add, the more we lose sight of the most important R, which is replacement. You can carry on reducing, you can refine the experiment, but if you are not replacing the animal, you are still inflicting torturous experiments on those animals, and it's not right. This year also saw the introduction of the Rawle report. Now, in the UK, we have the National Centre for 3 R's, and they conducted an independent review by Dr. Francis Rawle. And what they found was the replacement doesn't seem to be covered well at all by any of the review processes. The people who are in power, the animal welfare and ethical review bodies and the animal and science regulation unit inspectors, have no understanding of replacements and so rarely suggest it. And that means that there is just a free pass of project licenses for animal testing, because if they don't know how to scrutinize it, why are they getting through? And I'd like to bring you to this as well. Now, some of you might have heard of Doctors Against Animal Experiments, but they're a group based in Germany who looked at the public funding money in Germany, and they found that less than 1% was done on non-animals, was done on cutting edge human relevant, and more than 99% was done on animal research. The 3 R's are not working because we are still using the rest of the R's to justify animal testing, and it's completely not okay. And they also introduced another concept that you might find interesting, that maybe we should get rid of the word replacement. Because the word replacement means that we are comparing it to animal validated data, and that animal data is the gold standard. And we know it's not the gold standard. Go back to the start of my presentation. Look at the number of times we've harmed human life. We've harmed humans and animals alike. So really, maybe we should start using words like animal-free innovation, cutting-edge technology, and start thinking about that real paradigm shift of our language. And when does it simply just become animal abuse? Now, if you looked at Charles River's criminal record sheet, it'd be astounding. They violated Federal Animal Welfare Act several times, from inadequate veterinary care to failing to provide suffering animals with any sort of pain relief, baking alive monkeys when they're the thermostat malfunction and no one noticed. There is no proper care and animal welfare being done to any of these animals, and we are still letting it happen. And we need to be able to shine a light and say very clearly when will this barbaric animal torture end? And recently, look at the PETA exposés that have been done. Charles Rivers have been implicated, allegedly, in monkey trafficking, and not just from Cambodia, from other countries as well, Mauritius included. Now, I hope I've enlightened you in some way, because without enlightenment, there can be no revolution. And friends and colleagues, the revolution is already upon us. In vitro tests, which you'll hear about over the next 2 days, they will, these animal-free innovations, they will be able to sufficiently predict drug responses in humans. And if we have better predictions, it means we have better efficacy and safety. And now you're going to ask me, how do you get the data from humans before you've even started human trials? Now, the healthcare industry is rapidly moving with technology as well. We now have the infrastructure for cloud computing, and with being able to have these kind of electronic health records available and this wealth of data, we can provide better deep learning models. And so the better data we have, the better we can predict drug responses within humans. And I want to bring you to, I was reading about this AI system called Biosim, and what they did was they created a digital twin, right? So a digital version of the human body, so that they could identify how medications behave, whether it's injected, taken orally, or via the skin. Every hundredth of a second, the AI system simulates how drugs interact with different parts of the body to produce vital drug information, such as metabolism or organ toxicity. Now, without using any animal testing, the method provides forecasts in a matter of seconds and has been proven to be more effective than any animal testing could have been. It's faster, cheaper, and more effective. And also virtual reality. Now, some of you may have used virtual reality to play games at home, but also it is changing the way in which we teach medical and veterinary sciences. In 2016, the University of Chicago Hospital replaced all animal testing. So what they used to do was use pigs for practicing surgical training operations. And now the whole thing is done via VR. Even the US Food and Drug Administration is finally recognizing that AI will be responsible for ensuring that safe, life-saving products are available at a faster and more accurate rate than animals. It is fantastic how far we can get. And finally, you're going to hear a lot about probably organ on a chip and, the rest of these 2 days. But I'd like to draw your attention to, it was last year that they found that, organ on a chip models improves preclinical toxicology much more than, say, what a traditional animal model was, what was used for. We can apply deep learning approaches to 2 very key areas, human on a chip and rare disease on a chip. Now, a human on a chip may involve being able to aggregate multiple, a combination of different organs on a chip and be able to provide, with these deep learning approaches, be able to provide accurate data on drug response, whether it's through growth, differentiation, metabolism, etc. And finally, with problem with rare diseases, we have such a small patient pool. So drug discovery can be really difficult. And so that can be scaled, and we can introduce targeted mutations with a rare disease on a chip in order to screen for drug toxicity, but also drug discovery. Harmful practices over the years had to be abolished. And the example I've given today is the Black Death or plague. We had to abolish these shameful practices. And animal testing is no different. It has no place in the 21st century. Now, when you look at the worldwide animal testing numbers, it is a sobering thought. more than 150 million animals worldwide in lab experiments every year. Who suffers? Because I've talked about it earlier, it's not just animals, humans suffer too. When will our suffering end? And when will we finally realize that we need to bring animal-free research to the forefront? The future is medicine without cruelty. And thank you very much. I'll be more than happy to take any questions you have.
Open for questions if anybody has. Well, if no one has a question, Kavita, quick one from my side.
Absolutely.
Very thought-provoking presentation.
Thank you.
And while I was listening to it, and one of the things really struck me was that, you talked about VIOC, you talked about that, proven human toxicity despite having clean animals. Okay, and cardiotoxicity and, respectively. But can we have, now with the development of many alternate models, can we get them tested in these alternate models, show that they are really toxic, and that will really demonstrate that, alternate models are much better than ours?
Absolutely, and I kind of wish I brought a a PDF of all the papers that we've put together through Medicine Without Cruelty. Now, the one that I was talking about earlier about the liver organ on a chip, they tested it on drugs that were known to be produce a toxic effect, and what they found in animal models is that it didn't have any, it was proven to be safe. And so we've already improved the preclinical toxicity testing through, say, one liver on a chip. We can amalgamate, say, for example, you talked about cardiotoxicity, and we can produce that information and perhaps couple that with, for example, AI and provide that data readily available for us. Yeah, absolutely.
Yeah, we have to develop a lot of databases and we have to create
100%.
and so much variability is there, especially in case of humans. So many factors, in fact, see that some toxic chemicals, they act differently at the low altitude, and once their toxicity is different at the high altitude. And a lot of such variations are there, temperature variation. In case of fish, we see that there is a variation in toxicity level, if it is hot or it is cold, different. So what do you feel that, what is the future when every types of permutation and combinations are in the AI mode?
That's a very good question. And I think really what it's going to come down to is data sharing. What we need to have is, from a regulatory point of view, and, perhaps you'd like to chip in, but being able to bring together and open up, say, like I mentioned before, healthcare industry data, we can then use that to further, solidify the databases that we have available. But yes, you're right, an expanding field, and there's lots to be done, but we need the investment there now.
May I ask one question,
Absolutely.
First of all, I would like to say very, thank you very much for coming here, and very good morning to you. My question is, how do you define the Australia's increasing farm cruelty on the animals like chickens, porks, and like cattle? Like we are trying to do decreasing the rate of using animals in the experiments, whereas Australia is increasing their farms, and that is becoming a major factor to cruelty to animals like chickens and porks and all. Like one documentary also is there, like Dominions, So it has revealed what is actually the Australia is doing against the animals' rights. Thank you very much.
Well, it's a fascinating discussion, definitely. Not something that I've ever been a part of, but I know that there are plenty of animal welfare organizations. So I don't know whether you've heard of Humane Research Australia. That's, led by a good friend of mine, CEO's Rachel Smith. And, the work that they're doing there is truly incredible. But yeah, definitely something that I'd love to pick up with you further.
So you'll be interested in knowing how Savita has been brought to India. I met Savita World Congress on Alternatives that was held in August 2023, Niagara in Canada. Among the many people I met, I had a lot of passion for the kind of approach Savita has been developing, and the openness, the frankness, and the ability to respond to the developments in the arena of alternatives. I was really fascinated by her interest in this topic and her contribution and the passion. So when decided to host this conference here, I suggested to Zia that she may be invited as one of the speakers from abroad. And among the many people who agreed to participate in this Congress, in this meeting, at least 10 of them was interested in joining, but they had reasons not able to really come to India. So they will be addressing the conference through online mode. Savita was very nice to have found time and money to travel all the way from UK to India and not only spend the days, but spend money and share her very interesting thoughts. I'm really very much impressed by the thought sequence she has been developing. She is a little ahead of all of us. That's what I would say. We have certain passions, like starting from the 1959 Russellenberg 3 hours. We have been passing through that tradition and ultimately to see if replacement could be the ultimate answer to all the issues. She has gone much further to say that it doesn't stop there, but don't even trouble the humans. Don't even trouble the humans and try to do things in the models which are close to humans, not animals and not the humans. Do it in the alternative models which are close to the human and through genetic analysis. That's really very fascinating. And some of the expressions she gave has been really very impressive. And I thank especially Savita Newton for accepting our invitation and making it possible to come to India and deliver a brilliant lecture on a newer theme, a concept which can be carried forward much. And of course, she has the age, she's young, and she's now heading an organization, an NGO organization, and therefore she should be able to carry her thoughts forward and give a translational effect to that, so that in course of time, genetic approaches can also be a component of the alternatives domain. Thank you, Savita, for your wonderful presentation, and we really enjoyed it. Thank you so much.
Thank you, ma'am, for this wonderful session. Now I request Professor Dr. M.A. Akbar Shah, sir, to hand over the memento to Dr. Savita Nuthan, ma'am.
Now I request Dr. Adeep Roy, sir, to present a memento to Professor Dr. M.A. Akbar Shah, sir. I request Professor Dr. M.A. Akbar Shah, sir, to present a memento to Dr. Adeep Roy, sir.
Thank you, sir. Thank you, ma'am. Now I would like to invite Dr. Hiramani Rabha and Dr. Ajit Devia to host the next section.
Good afternoon, everyone. I, Dr. Hiramani Rabha,
And I'm Dr. Ajit Sevier.
Junior Residence, Department of Pharmacology. We are honored to introduce Dr. Ahmed Nazmi sir and Dr. Gigi Ahmed Sadab sir, Associate Professor of Genetics and Department of Zoology, to be the chairperson for this session.
Dr. Ahmed Najmi sir, Associate Professor, Department of Pharmacology, AIMS Bhopal. Sir is an alumnus of JNMC AMU. Sir is the author of book EC Concise Goal-Oriented Pharmacology. He has published papers in the reputed journals, including the Journal of Pharmacology and Pharmacotherapeutics, the International Journal of Medico-Biological Research. Sir has been awarded with the Uppsala Prize for the best poster presentation also. Sir has been is the life member of Indian Pharmacological Society and Ibn Sina Academy of Medieval Medicine and Sciences, Aligarh.
Now I would like to request our chairperson to introduce our speaker, Dr. Adrian Smith, for this session, who will be delivering his lecture online.
A warm welcome to all respected delegates. I am really privileged to be invited here to chair this session, and I am especially thankful to Dr. Syed Ziaur Rahman, who is the organizing secretary of this international conference and also my teacher during my undergraduate days. Now, I would like to invite our next speaker, Dr. Adrian Smith. I am honored to introduce Dr. Adrian Smith, a distinguished British Norwegian scientist whose illustrious career has greatly impacted the field of veterinary medicine and laboratory animal sciences. Sir earned a doctoral degree from the Norwegian School of Veterinary Science, and with a career spanning over 4 decades, Dr. Adrian's dedication to animal welfare extends beyond his academic and research roles. Sir has been instrumental in organizations such as Norcopa, the Danish 3R Center, and the Danish National Committees for the Protection of Animals used in the research. His contributions have been widely recognized with prestigious awards, including the Nordic Prize for the Alternative to Animal Experiments, the Sir Patrick Moore Award for the contribution to animal welfare science, the Scandalas Award of the Honor, and the Charles River Excellence in Refinement Award. Now, I would like to invite our speaker, Dr. Adrian Smith.
Thank you very much for your kind introduction and the invitation to speak at this important meeting. I must apologize for my voice. I have a bit of a cold, but I hope you can see me and hear me okay, and that you can see my presentation on the screen. I have quite a lot of slides, but you can download all the slides from the address on the screen. That's noricopa.no/SAAE-I. I'm a laboratory animal veterinarian, and I've been working for many years to try and improve the way we use animals in research and implement all the 3 R's, that's replacement, reduction, and refinement. I've been secretary of Noricopa since it was started in 2007, and our aim is to help scientists to find and disseminate information about all these 3 R's, and I will come back to how we do that in a moment. You're very welcome to contact me after this meeting if you would like further information using the email address on this screen. Our work is designed to promote better science, and what do I mean by that? Well, if we can replace animal use entirely, then that is our most important aim. But we live in a world where there is still animal use, and as long as it's not possible to replace everything, then we must work hard to reduce the number of animals and to refine the use so that suffering is limited to the very, very minimum, preferably eliminated altogether. I also think that the production of valid data is an important part of science, that we make sure that as long as animals still are used, that the data we are getting from them is valid, that it's a true effect of the treatment and it's not caused by, for example, stress of the conditions under which these animals live, which is why it's really important to think of refinement as well as reduction as long as animals still are used. We have to make sure that the experiments are reproducible and translatable, and if we can't achieve that, then animal research certainly is not acceptable. We must aim for best possible animal welfare and also think about health and safety, not just of ourselves, but also of other animals that are in the neighborhood of the animals being used in research. And an important part of this is what we call a culture of care, which I'll come back to, making it possible to discuss openly and honestly the possibilities to replace, reduce and refine animal research, and communicating this best practice to others. We are not an organization just for scientists, or we are not an animal welfare organization. We are a consensus platform for all the major stakeholders around animal research. We are part of ECOPA, which was founded to recognize national consensus platforms where all these four stakeholders are equally represented. So we aim to advance the 3 Rs by getting researchers, regulators, people working in industry and animal welfare around the same table and discussing how we can advance the 3 Rs, but with consensus and agreement between these parties to improve understanding and avoid the unfortunate situation, which is often the case, where those who are opposed to animal research and those who are using animal research don't have good dialogue and explain their views well. So we work for all the 3 Rs of Russell and Birch, and I thought I'd just mention that we've recently produced a 40-slide PowerPoint presentation about the 3 Rs to explain all the concepts involved in this, which Russell and Birch developed during the 1950s. And this is available in 3 language versions, and you can download it free of charge from our website, and you can use it in your own teaching. What we spend a lot of time doing is maintaining and updating a website which aims to be an overview of global 3R resources to help those who wish to replace, reduce and refine animal experiments. There are about 10,000 web pages that get about 1,000 hits a day. And in addition to updating this website, we produce detailed newsletters 7 or 8 times a year with the latest advances within the 3Rs to help inform those who are trying to advance the 3Rs. And I'll come back to the newsletters at the very end. There are now an awful lot of online meetings and physical meetings about the 3Rs, all aspects of them, and we maintain a very comprehensive meetings calendar. Many of these events are recorded as well, so we have other web pages where you can identify meetings that have already taken place but which have been recorded. So if you missed a meeting, you can often gain valuable information by listening to a recording. And these have been sorted by the topics in the prepared guidelines, which I'll be coming back to in a minute. I just thought I'd mention quickly that there is a very good course on ethics and design coming up very soon, It's an online course, it's very cheap, and if you download these slides, you can get the information about how to register for that. It's organized by a group of teachers in Nottingham in the UK, who I can highly recommend. The NORICOPA website contains a lot of databases, and I'll come back to some of those in a few minutes. And it also has links to over 70 other databases, which contain information about replacements and to animal experiments and ways in which we can reduce and refine those experiments which still have to be performed. One of the databases within NORICOPA's website is called 3R Guide, and this is a collection of over 400 guidelines for how to implement the 3Rs, and we've collected them from all over the world in collaboration with the USDA National Agricultural Library and Tim Allen's work in particular. We have also a database called TechSpace, which contains information on about 1,500 books related to laboratory animal science, to animal welfare and alternatives to animal experiments. And we have a database called Norina, which contains information on about 3,000 products that can replace totally or supplement animal use in education and training. This is at all levels, from dissections in schools through undergraduate training in, for example, physiology and pharmacology, where we can replace the classical experiments that have been used there, and also through to the needs of animal technicians and scientists who still have to use animals, but who need to learn techniques like, for example, blood sampling or injection techniques or surgical techniques, which they can practice on simulators and mannequins to learn these skills before they actually approach a live animal. This infographic was produced by Utrecht University in the Netherlands recently, showing the pathway to better science. And we are also working hard to promote this pathway and to promote openness and share information about alternatives. And at the beginning and end of this pathway, there were two sets of guidelines, which I will be talking more about. The PREPARE guidelines, which we have produced ourselves, and the ARRIVE guidelines, which are guidelines for reporting animal research that have been produced by the NC3Rs and others in the UK. And I will explain how these can be used to advance these 3Rs. Sometimes we hear that the way to improve research is by better reporting, but I do believe that it's really important to start at the planning stage. Here's a picture of a burnt cake, and however well I describe that cake, I can't improve on it. The fact that it's burnt is nothing that I can do anything about by describing it better. I have to go back to the kitchen and start there by changing maybe the baking time and the ingredients, which is why it's really important to begin by preparations from day one. And we have looked to other industries who are very good at precision, replicability, and safety, and seeing how they work. And we see that checklists are a very important part of their work. How even on a short flight, pilots will use 10 or 15 checklists to ensure the safety and quality of their flight. And these reduce the risk of them forgetting to carry out important actions, make sure that everything is done in the correct sequence, encourage cooperation and cross-checking between crew members, and make sure literally that everybody is on the same page. And the PREPARE guidelines, which we published in 2017, are an effort to tell scientists how they should increase this dialogue, how they should be aware of alternatives in the broader sense to animal research, and how they should prepare for research that looks as if it might involve the use of animals. And these have been quite well received. This paper has been viewed or downloaded over 30,000 times so far. Prepare consists of 15 main topics. The first set of topics is about formulation of the study, and the very first part of that is about the need to go to the literature and find out whether the proposed use of animals is necessary at all, whether there are replacement techniques that can be used, whether it's possible to synthesize evidence from work that has already been done instead of starting a new experiment. And checking, of course, the legal issues and the ethical issues. Is this experiment acceptable after a harm-benefit assessment? If it has to be done, can the humane endpoints be put in place so that the animals don't suffer? And only after all that's been done, if it's still concluded that animal research has to be done, then looking at the experimental design and how to analyze the data coming from it. And then the second important part of Prepare is about a dialogue between scientists and the animal facility, making sure that all the practical details are being considered, the time scale, the funding, the division of labor, evaluating the facility to make sure that it is actually possible to do the experiments that have been planned at the facility, that the staff have the necessary education and training, and the health and safety aspects. Are there any special health risks, and how are we going to dispose of any waste produced during the experiment and decontaminate the rooms that have been used? So the first stage, as I said, is all about checking whether it's necessary to do the research at all. The second important stage is dialogue between the scientists and the facility where it's going to be done. And then finally, an important section on quality assurance of the substances, the procedures, the animals, and the conditions under which they're going to be kept, the procedures themselves, and then what happens to the animals afterwards. Whether they have to be humanely killed, can they be released, can they be reused, or they can be rehomed. And here I've highlighted all the aspects of Prepare which are not typically mentioned in reporting guidelines, to show how it's important that we have both guidelines for planning animal research and also for reporting it, because there are a lot of crucial steps which don't get reported in scientific papers, but which will affect the quality of the work being done and the animal welfare. Prepare consists of two parts. The first is a checklist. You can see on it's a 2-page checklist. The 15 points have also a number of subtopics, and it's available at present in 35 languages. And there are also 2 online versions in addition to this simple PDF file. So this can be used by scientists from day one of planning, when they're first starting to plan their research, which might involve the use of animals on paper, and they need, as I say, to go through the points I just mentioned, including literature studies as the very first stage to see whether it's actually necessary to do the experiments at all. But it's a checklist they can use to make sure that they really have considered all the aspects that one should consider when planning research that looks as if it might involve animal use. In addition to the checklist, there is a lot more information available on the NORICOPA website, and we have pages for each of the topics and subtopics on the checklist, and these pages include links to quality guidelines and scientific papers from all over the world. on the techniques that will have to be used if they're still using animals, but of course also links to databases, scientific papers, and other sources of information about alternatives to animal experiments. And those who are using fish in research will also find that there are additional.
बाद में मैंने सोचा यार मैंने उनसे बिना पूछे ही दे दिया, पूछ तो लेती कि क्या है, किसने दिया, किसको चाहिए।
There are also separate tabs with information for those using fish in research. There is also an important part about what we call threat and error management. That's making sure that we try and plug all the holes in the Swiss cheese, that is the weaknesses and the dangers that are often associated with research, making sure that we cover as many of these as possible to avoid serious incidents occurring. This is an important part of any practical experimental work. And if incidents do occur, then there is a portal called SAAE-I, which is very good for reporting accidents so that other people can learn about these. And they have a website where you can anonymously report accidents that have occurred, and these are analyzed by a team of experts who then comment upon how this could be avoided in the future, and then they publish this information on the website so that other people can learn about the accidents that occur and avoid them happening again. We have also launched a refinement wiki. This is somewhere where scientists and technicians can quickly publish information on how to improve procedures that are still being used in animal research to make sure that we reduce suffering to an absolute minimum. And this is rather like Wikipedia. It's somewhere where you can add text and links, pictures and videos. And we have about 60 or 70 topics at the moment. So this is one way in which we're helping the scientific community to discuss the areas of animal research where they feel are particularly controversial or where there's a great need to refine and reduce animal use. We also encourage the culture of care in all scientific institutions that are still using animals. We are the hosts of an international culture of care network. And culture of care is all about showing that the whole establishment from management downwards is committed to improving animal welfare, to improving scientific quality, to taking care of the staff, and being transparent to all stakeholders, including the public. And this network shares experience on how we can improve a culture of care, and has a lot of resources with examples of events that can be held, how you can increase communication. The idea being that we want anybody at an institution where animals are still being used to feel that they have the confidence and the respect to voice their concerns or their opinions about the way things are being done without any risk of reprisals or ridicule, so that we can improve the standard of the work that's being done, advance the 3 Rs in an honest and open and caring environment. And if we have this type of culture of care, then this facilitates honest discussion, and important questions about animal use can be asked in an open and constructive and friendly environment. So if we hear comments like this, well, we've always done it this way, or we do things as often as necessary, or there are no alternatives to animal use, then alarm bells should ring, and we should start discussions about whether it is actually possible to change the way in which we're going. And closely related to a culture of care is what Susanna Lewis has called a culture of challenge. That is, that we look for what is acceptable rather than choosing what's been accepted over the years. This is a really important principle if we are going to advance the 3 Rs and ultimately replace animal use. The Culture of Care Network has representatives from a lot of countries, not yet India. We'd be very happy if representatives from your country would like to join this network. This is taken from an interactive map that Noricopa produces, and we have a lot of information on that map. For example, you can find centers worldwide for replacement, reduction and refinement of animal research, and information on a lot of associations that work worldwide to advance the 3 Rs. And if you have an organization that would like to be on this map, then do please let me know. So now let's look a bit quickly at the other end of this pathway, the ARRIVE guidelines, because the way in which we report experiments that have used animals is really important, and it's an important part of the work of evaluating how we can in the future replace that animal use. The ARRIVE guidelines have been produced in 2 versions now. The latest version was made because there was not enough compliance with the first version of it, so it's been split into 2 parts now. The ARRIVE Essential 10, which is a list of 10 items that are the basic minimum that should be included in any manuscript that is published and describes animal research. And then the recommended set, which is another 11 items which add important information. But since the level of compliance has been disappointingly low for the ARRIVE guidelines by journals, then they are trying to get full compliance with the Essential 10 list first, and then hopefully a bit later the recommended set of 11 will be complied with as well. And the ARRIVE guidelines have a website that looks very much like the Prepare website, where they have text and links to more information on each of the topics, just as the Prepare guidelines have. And we have also made sure that there are links from the Prepare guidelines to examples and references from the ARRIVE guidelines. So these two are very much interconnected on the Noricopa website. And the idea of this is that it will be easier for scientists when they submit their manuscripts to journals for publication to find that they have answered or thought about already the questions and the comments which the reviewers and the journal editors have, which means that it will be easier for them to get their papers accepted, which avoids a waste of human resources and animal lives, which is the case if the papers are rejected. So by starting on day 1, by using the Prepare guidelines to decide whether the experiments need to be done at all on animals, and then working through all the topics on Prepare and keeping an eye on the requirements in the ARRIVE guidelines as well, then we should see better science without animals where possible. We should see improved animal welfare where animals still have to be used. We'll see advancement of the 3 Rs and a safer working environment as well at the facility. And this will produce better cakes as well, to use that analogy that I mentioned earlier. The 3 R's were proposed by Russell and Birch in the 1950s to reduce harm to animals. This was their primary intention. And whenever we're planning animal experiments, we have to look for ways to reduce harm and increase benefit. And the 3 R's were mainly about minimizing harm by replacing unnecessary experiments, reducing animal numbers, and refining conditions. But there's another important concept which I'm very fond of, which isn't nearly as well known, called the 3 S's. And these were proposed by Carol Newton in the 1970s. And they are good science, good sense, and good sensibilities. Good science is what everybody is aiming for, of course. Good sense, I think by that she meant use your common sense. If an experiment that is being planned doesn't make good sense, then it should be re-evaluated, adjusted. And good sensibilities is all about using your heart as well as your head when planning research. If, for example, a procedure that's being planned would be unpleasant to us, then we should always give the animals the benefit of the doubt and assume that it would be unpleasant to them as well, and either avoid doing that procedure at all, or making sure that the animals are given adequate pain relief during the experiment. And I said that it's important to make sure that the data coming from the experiments is valid, and the 3 V's is another important concept now. Construct validity. Can the model answer, in fact, the question that's being asked? Internal validity, has it been correctly designed? And external validity, are these results translatable to the target group, for example, from animals to humans? And we have more information on each of these 3 concepts on the pages at the bottom of the screen. I don't have time to talk in detail about alternative methods now, which is an enormous topic, but I would just like to clarify what we mean nowadays by alternatives, and they're often called NAMs and NATs. NAMs are new approach methodologies. These aren't direct replacements for the use of animals, but they are techniques which avoid animal use. For example, the so-called read-across methods, where we predict the activity of a new chemical by comparing its structure with the structure of known chemicals of which we know their activity, and then predicting the activity of the new ones from by that means. There are methods like studies on organs like the human placenta, where we're not replacing animal experiments, we're actually just using human tissue as our starting point. These are so-called NAMs. And then we have NATs, the non-animal technologies, where we are actually replacing animal use by, for example, organs on a chip, or organoids, or experiments on animals that we don't consider to be sentient, like fruit flies. And it's important that we spread information about all these alternatives, and this is one of the reasons why NORICOPA has built this large database. And it's important that we have dialogue between those who are using these methods and scientists using animals. Many of those, for example, working on NAMs may not even be aware that they're using methods that can reduce animal use. And it's really important that we build bridges between the laboratory animal community and the communities working on these non-animal methods. So we need a lot more dialogue between the experts in their different fields. And this is one of the reasons why NORICOPA believes in the concept of dialogue and consensus, that instead of just shouting at each other. We try and get around the same table and discuss specific ways of replacing and reducing animal use, because we all have different expertises. Those working in the animal house know a lot about the animals and the way in which they're affected by their environment. The scientists know a lot about their area of science, but maybe not enough about the animals that they wish to use in their experiments. And we need to bring these groups together if we're going to advance the 3 Rs. To try and advance this work further, we produce, as I said, very detailed newsletters 7 or 8 times a year with the latest advances on the 3 Rs. And I'd be very happy if you'd like to subscribe to those. They're in English. They are sent out internationally. There are about 1,300 subscribers at the moment. And I'd like to thank our main sponsors for the work of Noricopa, and tell you that you can subscribe to these newsletters by using this box at the bottom of each page on the Noricopa website. You can add your email address there, press the register button, and then you will start receiving our newsletters. So I'd like to thank you for listening and remind you that these slides are available at this simple address, noricopa.no/SAAE. I'm sorry, there's a spelling mistake here. It should be SAAE, of course, -I. And I'm very happy to take any questions that you might have.
Thank you, sir, for this wonderful session. Now I request Dr. DJ Hamad Ahmed Shadaf, sir, to give this handover to token of appreciation to Ahmed Nazmi, sir.
Okay. Thank you, Dr. Adrian Smith, for your enlightening talk on how to use 3 Rs for betterment of science. Now I open the session for question and answers. If any questions and answers. Okay.
As I said in my course, I'm very happy if anybody would like to send me an email. I'm very happy to answer questions and afterwards. So it's no problem at all if nobody has any questions in the audience now. I'm very happy to receive emails from you later.
Okay. Thank you so much. So we can proceed the session further.
Now I request Dr. Ahmed Nazmi, sir, to hand over token of appreciation to Dr. DJ Hamad Ahmed, sir.
Thank you, sir. Next, we will gather for the inaugural session after a break of 15 minutes. Thank you.
हाँ